Today we will be continuing the lecture on lipids. So what happens when your body processes triglycerides? What do you get? So here we have the triacetylglyceride. We're going to call that TAG. Okay, so in some textbooks, you might have this abbreviated to TAG. And all that means is triacetylglyceride or glycerol. Okay, the acetyl comes from the 3 carbons. That's just shorthand. Okay, so triacetylglycerol or glyceride. So you have that, and your body wants to use it for energy. Okay, so you have some water, and you're going to have a lipase. Now, lipase is actually an enzyme. Okay, so it's an enzyme. That's a really cool Z. It's an enzyme that actually breaks down the triglycerol. Okay, and you're going to have some energy whenever you do that. And what you get out of that is that you're going to get a glycerol. You're going to get a glycerol. And you have 3 fatty acids. So here's an FA right here. So we have 1 FA, 2 FAs, and 3 FAs. Okay. So again, that's kind of like this right here. We're just going to have some random amount of carbons. Okay. So that's what happens whenever you break down a triglycerol. Again, the enzyme that does this is the lipase. Okay, so you can imagine lip as fat and then ase as the enzyme. Okay, so you can really think of it as the fat enzyme. It's the enzyme that breaks down fats. Where is this enzyme located that you may need to know for an exam? So this is commonly found in the intestines. Found in the intestines. And, you know, that makes sense because if you go to McDonald's and you get the, I guess, Travis Scott burger, it's just a normal burger. I don't know what's the hype about, but let's say you get that hamburger. Well, when you eat it, it goes down to your intestines, and your intestines begin processing it. So it starts taking all the nutrients and the fats with it. Now, the fats in the food will be digested by lipase, so be processed, by lipase, and you'll have some energy. Okay? So, while we're in the subject, where do all the fats go in your body? Like, where are they stored? Are they stored in your fingertips, in your hair? Where are they? Well, they're actually stored in small little cubby holes, okay? Like small little cubby holes that I call adipocytes, okay? So, adipocytes store fat. Okay, so imagine when you were in kindergarten, you had this furniture that had little compartments in them, and those were called cubby holes, right? So you can imagine these cubby holes as adipocytes. The little kindergartners, they go into the room, and their backpacks are fat molecules. So they go to the adipocyte, the cubby hole, and they put their backpacks in. Similarly, the molecules bring the fat into the adipocyte, and they store it there until you need to use it. Okay, like when you're using energy for exercise, your adipocyte will release the fat that was stored. Okay, so this is very important to know. So this is important. Okay, so these two. We will now be talking about saponification, right? So this is saponification. Okay, so what is saponification? Well, it's actually when you treat a fat with a very strong base. For instance, we're going to be treating this fat with sodium hydroxide. Another word you can call sodium hydroxide would be lye. Okay, if you remember, back in like the 1930s, people used to take showers with lye. They would use it in shampoos and soap and whatever, and it was really strong. It would actually kill parasites on your skin, like ringworm and some other stuff. But if you got on your eyes, you would go blind. You had the potential to go blind. So we don't really use lye in soaps anymore. However, we do use it in the process of making soap and pretzels, actually. And lye, if you eat it by itself, is very, very poisonous. But for some reason, bakers use it in pretzels. So go figure, right? It's weird. But what happens is that you get this fat, and you treat it with a very strong base. Now you're going to cleave the fat to get the glycerol, okay? So pretty normal. But instead of just having the fatty acids, you also get an ion, okay? So you kind of have a salt. You have a salt. So these are salty fats. Let's call them salty fats, yeah? And these are quite yummy. Of course, that's a joke, right? You don't eat this. So this is actually pretty safe, and it forms like a solid that is kind of neutral in color, and you can add food coloring, pigments to it or whatever to get the color, and then you can add some herbs to get like the smell or whatever. But it's very soapy, it's basic, and that's saponification. Okay, so you may see this on Instagram or social media of people making soaps or carving soaps. That's just a product of saponification. Okay, so this is important to know. Saponification is just the addition of a strong base to a fat to create a soap, quote unquote soap. Okay, so soap is just the salts. So sodium salts of the fatty acids. If on the exam they tell you what produces the sodium salt of a fatty acid, you say saponification. Okay, so that's all you need to know about that. Now we move on from triglycerides and we begin talking about phosphoacetylglycerols, also known as phospholipids. So this is a phospholipid right here. Phospholipids. Okay, so phospholipids, what are they? Well, essentially, you still have the glycerol, okay? You have the glycerol, but one of your fatty acids is converted into a phosphate group and an alcohol, okay? So this right here, this is going to be polar. Polar, and this right here are going to be non-polar. This is also kind of like non-polar as well. Okay, so this glycerol is, ignore it, okay? The main interest is the polar head, or the phosphate group. Now here we have the glycerol, right? So there's a glycerol, but notice that one of the fatty acids is not there. Instead, it was converted into a phosphate group, okay? Into a phosphate group. Now, why is it that this phosphate group is pointed away from this guy? Why do we not have a phosphate group right here? Well, it would be sterically hindered. Notice that this oxygen would be next to this oxygen, right? Or this phosphate group would be next to that oxygen. That's not favorable. You would actually spend more energy trying to keep it that way than you would if you kept it that way. So that's why the phosphate group is pointed away from the oxygen right there, okay? So that's why we consider this to be the head, right? So this is going to be the head. Let me just change that, head. And this is going to be the tails, okay? Now, phospholipids, let's call them PLs, right? So phospholipids tend to be about 16 to 20 amino acids. But you'll almost always see phospholipids within the range of 16 to 18. So you have 16 to 18 fatty acids. So that must mean that the minimum that you may see is about 16 carbons, and the maximum that you may see is about 18 carbons. So it's kind of like around that neighborhood. So very common. Very common range. Okay, cool. Now, let's talk about positions. So let's say that this is position 1. Okay, so you remember in the last video I said we're going to be talking about positions for phospholipids? Well, let's call this position 1. Now, position 1 actually favors saturated fats, okay? So position 1 favors saturated fats. Why is that? Well, saturated fats, they can be very compact and rigid, so they don't need a lot of space to function. Notice that we can have these two fatty acids right next to each other. They can hold hands and they can, be calm. They can relax, okay? So position 1 favors saturated fats because they don't take up enough room, right? So they're pretty good. But if I try to make one of them saturated if I tried to make one of them unsaturated, I wouldn't have enough room to do that in position 1. Instead, let's ignore this fatty acid right here. Let's ignore this guy. Let's pretend that the phosphate group is right here. And let's pretend that this fatty acid is right here, going this way. Well, if that happens, I can have a unsaturated fat right here, and then I can have an unsaturated fat on position 1. So we can say that position 2 favors unsaturated fats. Okay, so there we go. If you need help kind of figuring that out, we can have, let's do carbon, and then let's just kind of do like a simplified fatty acid, right? So we have carbon, carbon, actually let's do phosphate, and then we have a carbon. If I do unsaturated, and I do this unsaturated, that's okay, you know, because phosphate is between them and it provides enough room to do it. So that's why position 2 is going to work out for us. This cannot happen in position 1. If you have 2 unsaturated fats together, that's not good. It takes up too much room. So position 1 favors saturated fats, position 2 favors unsaturated fats, okay? Now, here's a cute little picture of the lipid bilayer. So imagine that we're in the cellular membrane of whatever cell in your body, right? Well, your membrane is made of these fats, okay? These fats. So right here we have a phosphate group. And they're kind of like little spheres, right? And this is going to be the non-polar part. So this is going to be the non-polar tails. And these are actually the fatty acids, okay? So fatty acids right there, okay? So right here we can say that these are going to be polar heads, all right? Right there. Now, it's important for you to know that we consider phospholipids to be amphiphilic, okay? Amphiphilic. So phospholipids are, not or, but are amphiphilic. So amphi is kind of like a root Latin word that means both, and then philic means love. So both love, that means that it is a polar and non-polar molecule that is pretty self-explanatory. We have a non-polar tail and we have a polar head. So we consider that to be a mixture of the two. Okay, so the reason why we consider that is because at neutral pH, so at neutral pH, this guy, the polar head, has a charge. The polar head has a charge, okay? And that allows it for polar things to not enter the cell membrane. So for instance, sodium ions cannot easily enter the membrane. They would have to do active diffusion, but that's a different topic for a different course. So the reason why we have a phosphate head and the membrane is to kind of prohibit things from entering the cell or from getting out of the cell, right? So it just kind of adds like a layer of protection. So you can do reactions to do that. So yeah. But did you know that phospholipids can actually be degraded? So whenever you're hiking, you always have to pay attention to your surroundings, right? You don't want to be attacked by a wild animal. Most importantly, you don't want to be bitten by a snake. So why are snake bites, venomous snake bites, so bad? What makes them harm you? Like, how does that affect your body? Well, over here we have the Indian cobra. So this is the Indian cobra. Okay. And what it does is that it's going to inject you with a phospholipase. So injects phospholipase, specifically A2. So all you need to know is that phospholipase can actually degrade. It can kind of rip apart the phospholipid bilayer in your membranes, okay? And that's not good. That's not good. Why is it bad? Well, whenever that happens, it essentially attacks the blood, and the blood is going to rupture, and essentially your blood is like a basketball, okay? And a rattlesnake uses phospholipase to kind of stab the basketball and explode it. So whenever your red blood cells are in contact with phospholipase A2, they actually rupture, and you start losing usable blood. Okay, so that's not good. And what is responsible for the blood rupturing is called lysolithiasin. Okay, so lysolithiasin, lyso means cut. That's responsible for your blood bursting. So that acts as a detergent. So we can say act as a detergent. Acts as a detergent. Okay. So first, it's going to attack your phospholipase. First of all, it's going to inject the venom. The venom has something called phospholipase A2. That's going to attack your fatty acids in your phospholipids. Afterwards, you're going to have kind of like a weird structure. So let's draw glycerol. Okay. And then we're going to have the polar head, so we're going to have phosphate. But instead of having like two tails, well, phospholipase makes it into a one tail, okay? So we're going to have a one tail. Now this structure right here is called lysolithiasin. Now this guy right here is a bad dude. He's going to go to your blood, he's going to pop them. And that is going to kill you if you don't get treated, okay? So that is how Indian cobras are venomous. Right? Okay, so now we will be talking about sphingolipids. So what exactly are sphingolipids? Sphingolipids, you no longer have glycerol. Okay, so the whole glycerol was taken out, and you receives kind of like a sphingosine. So this whole thing in blue, we will call that a sphingosine, okay? So glycerol is replaced by sphingosine, okay? Now, what's important to know is that it's always going to be on position 2. The fatty acid can always be found on position 2. So fatty acid is on position 2. Okay, and also sphingolipids are more amphiphilic than triglycerides. Okay, so more amphiphilic than triglycerides. Right? But let's talk about what this X means. What does this X mean? Well, this X can be a series of different things, okay? If X, so let's write, if X is H, so let's say that this is an alcohol, then this would be called, this is called a ceramide. Okay, that's called a ceramide. Now let's fix this E right here. Okay, let's fix that. But if this X, so if X is a sugar, let's say like glucose or something, then we can actually kind of write it as a, how do you say, cerebroside. So then it's called a cerebroside. So it's kind of cool. If you know Spanish, like I do, cerebro is kind of like a root word for brain. Okay, so this right here is going to be for the brain. And this makes sense because the brain, its first energy source is actually carbohydrates and sugars, okay? So that's one of the main reasons why you should keep some carbs in your diet. Your brain mainly uses glucose, okay? So if X is a sugar, then we call this whole structure a cerebroside. If X is a hydrogen, as it's most often is, we call that a ceramide, okay? But what happens if X is actually a complex sugar, okay? So if X is a complex sugar, so what I mean by complex sugar, I mean kind of like rings. So imagine a bunch of hexanes, okay? But those hexanes are sugars themselves, okay? So it's kind of like ringed sugars. If X is one of those, we call that a oligosaccharide, okay? So we're just going to put complex sugar. That's going to be called an oligosaccharide. So if you have an oligosaccharide on X, we actually call that a ganglioside. We call that a ganglioside. Okay. So one thing that you should know for this class is the presence of sphingomyelin. So sphingomyelin is going to insert itself and become a sheath for nerve axons. Okay, so sphingomyelin insulates nerve axons. Okay, and it's part of the myelin sheath. So you can tell that sphingolipids are kind of important in the brain, okay? These are very important within the brain. And they're so important that if one were to mess up, you can have a lot of diseases. So for instance, if you have too many gangliosides in the brain, well, you actually get Tay-Sachs disease. Okay, so Tay-Sachs disease. Let's write that here. Tay-Sachs actually stems from having too much gangliosides. These gangliosides build up along the nerves in the brain, and they build up so much that they cause death. Okay. So what else can happen? If you have too many cerebrosides, you actually get Gaucher's disease. Okay, so this is Gaucher's disease. Okay, and what Gaucher's disease is actually the enlargement of the liver and the spleen. You also get really bad bone pain and some anemia. Okay, so why do you have so many cerebrosides? Well, this is partly because of you don't have an enzyme that breaks down the cerebrosides. So what is that enzyme called? It is called, let's write it over here, it is called cerebrosidase. Okay, so you don't have enough glucose cerebrosidase. Oh, it's such a pain to say, but you don't have that enzyme, or you don't have enough of it, and so you have the kind of like the buildup of cerebroside within the brain. Well, not the brain, but within the body, excuse me, and that kind of causes Gaucher's disease. So Tay-Sachs is by the buildup of gangliosides in the nerves in the brain, and that leads to death. Is also genetic, and the buildup of cerebrosides is called Gaucher's disease, and that is because you don't have enough glucose cerebrosidase in your body. Okay. Now we will be talking about non-saponifiable lipids. Okay, so these are non-saponifiable lipids. Okay, so these guys right here, they can actually make steroids and they can make hormones. So can make steroids and hormones. Okay. Now, what's different about these lipids is that, if you notice, they have a lot of rings. So they have fused ring structures. Fused ring structures. Okay? And that makes it so that it is very rigid. So very, very rigid. Okay? One of the most important non-saponifiable lipids is cholesterol. Okay, so cholesterol is commonly found within animal membranes, so it kind of provides structure and rigidity to the cell membrane, right? So it allows it to keep its shape. Now, the reason why this is important is because it comes from the ring structures. If cholesterol didn't have its ring structures, well, the cell membrane would be severely weakened, right? So that's pretty cool. Another thing about cholesterol is that cholesterol is slightly amphiphilic. So I'm just going to put cholesterol as CHOL. Okay, so that's cholesterol. It's going to be slight, right? So slightly amphiphilic. Okay. And that is because, well, there's a lot of carbons and there's a lot of rings, but there's going to be an alcohol. So notice that these little guys have alcohols on them. So what makes it amphiphilic is the alcohol. So we're going to put OH group makes it amphiphilic. Makes, I guess you could say slightly polar. Okay, there you go. Now, the reason why we focus on cholesterol is because cholesterol can be a starting block whenever you're making steroids or whenever you're making hormones. So if a scientist wanted to make steroids, they would actually build cholesterol, and then from that building block, they can build whatever they wanted, right? If they wanted to make testosterone, they start with cholesterol. If they wanted to make progesterone, they make cholesterol. If they want to make estrogen, they make cholesterol. So on the exam, if it comes up, they can say, what is a non-saponifiable lipid that is the most important in making steroids and hormones? You say cholesterol. All right? So now we will be talking about the biological membranes. So this is biological membranes. So what is that? Well, essentially it's a membrane that has lipid bilayers, okay? And they're made of phospholipids, Sphingolipids, and some other stuff, okay? So you can say that this is made up of phospholipids. I mean, it has to be because it's in your membrane, right? Phospholipids, it's made of Sphingolipids. It's just made up of normal sphingolipids. And of course, cholesterol. Okay, well, the polar heads you're going to notice are going to be outside and they're going to be inside, right? So let's imagine that the whiteness of this board is water, right? So let's imagine that this cell is inside of a cup of water, right? What you're going to see, well, let's take a ball and let's cut it in half. What you're seeing is the internal, right? So you have the non-polar groups inside the ball. Outside of the ball, on the surface, the thing that is making contact with the water is the polar heads, right? So the polar heads are outside interacting with water. Okay, and then inside is going to be the non-polar heads. So non-polar heads, or tails, excuse me, non-polar tails are inside. So again, this is kind of like a callback to the hydrophobic effect. So this is the hydrophobic effect. And you can see how important this is to biochemistry. It's important in the formation of proteins, and now it's important in the formation of biological membranes, okay? So it's very important for you to know that. Now, within this model right here, it is not completely solid, okay? So the kind of like the teal color is not solid. It actually has the consistency of vegetable oil. So if you played around with oil, that's kind of what's inside of this membrane, okay? So it has that consistency. And we call this kind of like the fluid, we call this the fluid mosaic model. Okay, because in real time, these little tails are wiggling around, okay? And these polar heads are moving side by side. So this guy right here, let's see if I can find it. This guy right here, he may move over there. You know, he might be over here the next second. And this guy right here, he may move that way and be over here, okay? And so you can say that, you know, phosphates, or I guess lipids, they move laterally. So you can say lipids move laterally. That means that they move side to side. So imagine you're playing with a Rubik's Cube, but the Rubik's Cube is going to be spherical, right? So it's like a new one. It's very difficult. Well, you can actually move the lipids, the pieces, side to side. You can also move it up and down, but we're going to talk about that in a few moments. Let's just focus on side to side movement. Okay, so lipids move laterally side by side. So that's why they're called fluid. Now, what dictates how fluid the membrane is the type of fats within it, okay? So if you have long tails, let's say 20 carbons instead of 4 carbons, right? You're gonna have more firmness, more structure, more rigidity. So the longer the tail, the more rigid the membrane is. The shorter the tail, the more fluid it is. Unsaturated fats are actually more fluid than saturated fats, okay? So we can say that sat fats are rigid, you know. Long tails are rigid as well. That means they don't move as quickly, okay? They don't move. So, yeah, I mean, I think those are pretty cool things. Okay, but like I said, what happens if you want to move up and down? Well, if I want to move up and down, we're going to need an enzyme, okay? So let's randomly say, sometimes they're in random positions, not sometimes, but, you know, let's say that this right here, this purple thing, is an enzyme. And this enzyme is going to be called flippase. Okay? Flippase. And what it does is that when the lipid, you know, hits the flippase, is going to go downwards and go over here, this way. So here's what happens, right? You have a lipid is going to travel. It's going to hit this flippase. And whenever it hits it, it goes downwards, and it can continue going this way, or it can continue going this way, right? So all it did was it moved it up and down. Now, that takes a lot of energy, and it's not very efficient, so it doesn't happen all the time, okay? It takes tons of energy. Takes tons of energy. Again, this is a flippase. So scientists are very creative when they're naming enzymes, right? You know, if they had something that broke down hamburgers, they would call it hamburgase, right? So this is just called flippase, right? So this is very slow. So this is slow. Okay? So this is a slow process. Now notice that whenever you move laterally, meaning side to side, you don't need any enzymes. So this happens very fast. Okay, sometimes you can't even see it happen. That's how fast it is. Now, it's also important for you to know that, sometimes lipids have a preference for where they're at. What I mean by this is, on the surface, you might have a certain type of lipid. Let's just say it's a sphingolipid, right? Because it's polar. You're not really going to see glycerides on the surface, right? It wouldn't make any sense. But you would see more glycerides inside, over here, because it's non-polar, right? So what I'm trying to say is, on the surface of the cellular membrane, you have different amounts of lipids. You may have more sphingolipids than glycerols. You may have more phospholipids than glycerols. But whenever you go inside the membrane, the non-polar part, you may have more glycerols. You may have more non-polar lipids than polar lipids, right? So it's not random. There's actually certain percentages that you might find in the cellular membrane. But all you need to know for this class is that there's a preference within the cellular membrane. So for an example, if you have a question on the exam where it says, where would you find the most amount of glycerols in a cellular membrane? Well, I would say, hey, I would find it in the inner membrane where it's non-polar. Where would you find the majority of sphingolipids? Well, I would say on the outer membrane where it's interacting with water, because sphingolipids are amphiphilic, meaning that they are polar and non-polar. Where would you find the most phospholipids? I would say on the outer membrane of the cellular membrane. Okay? So there's a preference for lipids on certain regions of the cellular membrane. Okay, so that wraps up part 2 for lipids. It's a huge chapter, but hopefully you're understanding lipids more and more every day. I know that you'll do well in your exams, and I hope that you continue to study. Thank you so much for spending time with me and for learning with me. Remember that I love you and that you can do amazing on your exams. All right, please have a great day, and thank you.